Hi, Year 12, slightly different way of us having to do things with everything that's going on, but this is where that I used to teach A-level PE, so there's no reason why it can't work for our B-Tech studies as well. So what you're going to get is a video of me talking over a PowerPoint, which hopefully you'll find really useful. There's a chance for us to recap the ATP and the ATP PC system work that we did in lesson, and then also a chance for me to teach you for the first time the lactic acid system, which is the second of 3 systems for resynthesizing ATP. Use the video, pause it, stop it, replay it, make your notes the way that you'll need them. There's a couple of questions that I'm going to ask you, and then we'll follow up with some exam questions as well. But this is your information for you to have and use in the way that's going to work best for you. Some of you will watch it a couple of times over, some of you will pause it for breaks to do some reading, whatever needs to work for you. So, quick recap, if I just move that on. Remember, if we remember ATP stands for adenosine triphosphate, you'll remember that we've got, that means the 1 mole of adenosine and the 3 phosphate molecules, and that really importantly that is the only usable form of energy that's in the body. There's 3 ways that the body resynthesizes ATP. You remember that process of how that happens, and you've got some detailed notes of that in your work booklet and in your lesson notes. But the last thing we did in lesson was the ATP resynthesis via the PC system. So if this is our ATP here, we've got our 1 molecule of adenosine, 1, 2, 3 phosphates. You'll remember with the introduction of the enzyme ATPase, that's broken down into ADP, adenosine diphosphate, which is 1 adenosine and 1, 2 phosphates, and the breaking of the bond by that enzyme ATPase, breaking that bond to release that 3rd phosphate molecule, then creates energy. Yeah, now the system, the PC system that we talked about, how that energy is resynthesized, is how then phosphocreatine, this here, plays a role in reattaching the phosphate. And you'll remember the controlling enzyme here was creatine kinase. So when creatine kinase is introduced into the mix with phosphocreatine, it breaks down that phosphocreatine to create energy that binds the phosphate molecule that's been created to adenosine and give us ATP rather than the ADP. Yeah, so you should remember that from your lessons, should remember that from the notes that we've had and the questions that you've answered, and also as a piece of work that you're submitting for me currently around that. If we take that forward, one thing that maybe wasn't as clear in my lesson, which you really need to focus on, and the couple of bits of work that have come through didn't have this as a key focus. Phosphocreatine is only replenished when intensity drops below maximal, so drops to sub-maximal. What that means is we know that the ATP PC system only lasts for 10 seconds. You'll remember that from lessons. We don't get the resynthesis or the recreation of phosphocreatine, those stores don't get replenished until intensity drops below submaximum. So if you are continuing to sprint, you aren't going to be able to replenish your PC stores. Really important point. And then just 2 things for you to think about in your own learning. Creatine kinase is activated when ADP is detected in the muscle cells. There's a simple why question. So why is creatine kinase activated when ADP is detected? And making sure that you make the link, knowing that creatine kinase is the enzyme that initiates the breakdown of PC. If you're struggling with that, drop me an email and we'll go into that in more detail, but that should be pretty straightforward. So that's in your work booklet, this table. Really useful table for you to understand the advantages and the disadvantages of the phosphocreatine system. I've mentioned this period of 10 seconds. Yeah, that's a difference in where we're going to go next with the lactic acid system, as is this here with there being no harmful byproducts. So if we move through, you've also got a copy of that sheet in your work booklet as well. Yeah, so I won't spend too much time on that now. I'll do another video in the future which really goes into the details of the 3 systems in comparison to one another. If we move to the lactic acid system, I appreciate that that could initially look a bit daunting and a bit confusing, but just bear with me on this. So the lactic acid system is still anaerobic respiration. It's still occurring without oxygen, okay? And once, if you see here, once PC stores are depleted, that means once all of our phosphocreatine has been used up, which is around about 10 seconds, the body switches to glycogen as its fuel. Now, you'll have heard that terminology before, glycogen and glucose. You'll have heard that at GCSE Science. You'll be familiar with those terms. What you need to bear in mind is that glucose is stored as glycogen. So when you consume sugars or consume carbohydrates, they enter the body primarily as glucose. Now, the body can't store glucose. It has to be converted to glycogen for it to be stored. Yeah? But for the body to use glucose, it then has to turn it back. Sorry, for the body to use glycogen, it then has to turn it back into glucose for us to be able to get the energy from that. So let me just say that again. The body can't store glucose. It can store glycogen. So the body converts glucose into glycogen for storage, but then the body converts glycogen back to glucose for it to be used as energy. And you'll see how that happens here. So glycogen is stored in the muscles and the liver. Yeah, so glucose is stored as glycogen in the muscles and the liver. There's an enzyme called glycogen phosphorylase, which is responsible for glycolysis. So you will have probably heard that at GCSE Science before. So that glycogen phosphorylase is responsible for glycolysis. Glycolysis being the process where glycogen is converted into glucose. Once the body's converted that glycogen back to glucose, then glucose is broken down by an enzyme here called phosphofructokinase. Now you can use PFK. In an exam you will get marks for PFK, but glucose is broken down by PFK, by phosphofructokinase. And at that point, we get a yield of 2 ATP. So the energy yield of the body breaking down glucose with phosphofructokinase is 2 ATP. However, when that happens, we get some byproducts which are created from that. Okay? And initially, the first byproduct which is created is pyruvic acid. Now, the body uses pyruvic acid when oxygen is present. And we'll go into that in detail in future lessons. But just for now, just bear that in mind, okay? So when there is oxygen present, the body uses pyruvic acid. It utilizes that. However, at the minute, because we're in the lactic acid system, we're still at a point where this is anaerobic glycolysis. So this is taking place in the sarcoplasm of the muscle, and it's anaerobic glycolysis. It means it's taking place without oxygen. So pyruvic acid can't combine with oxygen. And what the body has to do is convert pyruvic acid into lactic acid. And that's done through an enzyme called lactate dehydrogenase, LDH. So when oxygen isn't present, there's no oxygen present, the body can't store pyruvic acid, that's a poison. It gets converted into lactic acid, which is the byproduct which you'll be very familiar with, and you'll have heard that before. When oxygen is present, then the body utilizes pyruvic acid as part of the Krebs cycle to create aerobic energy, but I will go into that in further detail. So hopefully what I've just said makes sense. Look back over that, make some notes around it, review it, and you can keep watching that back, and if you're not sure on anything, then please get in touch with me. I'm just going to show you in a different diagram, just so that you don't get overly used to one diagram. You've got here again, you've got glucose and glycogen, like we talked about in the previous diagram, just bearing in mind the transfer between the two, and the body's storing glycogen, storing glucose. Glucose then being present here, being broken down by phosphofructokinase at this point, which is giving us that 2 ATP yield, but it's also creating pyruvic acid. And you can see here when there's no oxygen present, pyruvic acid ferments and lactic acid is formed, and that's where you ultimately get to that point of lactic acid. There's no mention of the LDH in this diagram, so just have that one in mind. LDH, lactic dehydrogenase. the conversion of pyruvic acid into lactic acid. So we should be really clear that it's anaerobic, it's without oxygen, and this is the process of the body creating energy through glycolysis, through the breakdown of glucose and glycogen, which is being stored in the liver or the muscles, breaking that down and receiving an energy yield of 2 ATP. Okay, now just to help you with your understanding of that, when does the lactic acid system actually start? It starts when we've exhausted our phosphocreatine stores. So if you think about the continuum, we've got the initial ATP stores, which last for 2 seconds. You've then got your phosphocreatine system, which lasts for 10 seconds. And then once phosphocreatine, remember, has been depleted, and it won't replenish until we work to submaximal, the lactic acid system then steps in. Now, I want you to have these two things in your mind that are trying to understand why the system stops. So why does the system stop? If you bear in mind that idea of a reduction in pH, yeah, so the pH level reduces, or an increase in phosphocreatine. So we've got to a point where phosphocreatine is being replenished and the pH level in the muscles is reducing. So giving us the opportunity for the lactic acid system to be stopped being used and stop creating those harmful waste products. Finally, just want the bit to finish up with, sorry. Advantages, disadvantages, again, a really helpful table for you to have here, just to understand on one sheet. You've got this in your work booklet. Big advantage of the lactic acid system is it lasts longer. Okay, having said that, it doesn't last for a huge amount of time, 180 seconds, maximum 3 minutes. You can train your lactic acid system to last longer, but generally you wouldn't get beyond 3 minutes, perhaps even 2 minutes in most instances. Oxygen is not required, so it happens quickly. There's no delay in waiting for that energy to be provided. But disadvantages there, we know that lactic acid has a debilitating effect on muscle function, and we only get 2 moles of ATP. So for all of what we're talking about, the breakdown there, there's only an energy yield of 2 ATP. Yes, that's higher than the ATP-PC system, where we're only getting 1, but it's still only a relatively small yield. So what I'd like you to do is, this is in your work booklet as well, this task, if you apply the table above to a sport of your choice, a sportsman of your choice, and really look to explain the advantages and the disadvantages that they'd have as impact on your sport. So have it a practical application to a sport that you've chosen, please. And then finally, this is also in your booklet as well. I want you to describe the process of ATP being resynthesized. So I want you to go through that flow chart and make sure that you're able to really go into the detail of that and understand that. I don't just want that copied out from what I've given you, and use a practical example to apply each stage. So what I mean by that is have a performer that you're talking about using the lactic acid system and explain what's happening to that performer at each point. Okay, hopefully that's been helpful. It's a different way of doing things. Listen to it back. Pause it when you need to, read up on things you need to, ask me questions as you do, and then I'll follow it up with some exam questions for next week. All right, thanks very much.